Let me see. If you use your data today, you will know how much data you normally use. You will know it. Good morning once again. This is another DIY bioinformatics practical manual. Believing that you have, believing that you have know and learned what you're supposed to do as it regards the practical. This manual is going to introduce you to basic bioinformatics tools and how to use them for environmental research, including including identifying identifying microbes, exploring diversity, and understanding the ecosystem. Case study and demonstration. So what we are going to do today, we want to look at how can you get some kind of organism from dirty river and clean river as a bioinformatician. In this session, we are going to learn how to use BLAST, which is a basic local alignment sequence tools, to identify microbes in two different water sources. That is, from source one, a polluted water, which is going to be river A clean stream water in nature, which is going to be river B. And we will see how DNA sequence refill the earth of an environment. So by the time that one comes, we are going to use how can you use the blast to be able to compare between these two. So we are going to do that by the time it's come. So the first thing is we collect water sample from two river source. After that, we are going to extract the DNA and identify amplify 16S ribosomal RNA gene. Then after that, you are going to paste the DNA sequence into NCBI. So, and you are going to make use of BLAST. Then the next thing is you choose 16S ribosomal RNA from the database, and you are going to run BLAST on it. Then the last step, which is going to be, you will now identify the top matches. The top matches, those ones that align most, you are going to pick it in order to determine the microbial population in both clean and dirty water. So, and from what you are going to get from what is being done, river A results produces higher presence of bacteria, which is mainly pseudomonas, which can tolerate pollution, as well as indicate environmental stress or contamination. Then stream B, that is going to be of a more balanced and natural microbial population, which is going to indicate a more healthy and undisturbed ecosystem. Just like I said, the steps that we are going to use in performing blast is going to be refilled, and it is already displayed on the screen for you. Especially even if you don't have any, if you don't know what to do, you should be able to have it on the images. So a situation whereby you are going to have, you don't have a wet lab done, so what is going to appear there is just you take the NCBI query sequences. So I told you the sequences, we have query and subject. The one that you are going to use is always going to be query, the one you are going to get from NCBI, and the one that you are going to compare them with are called subjects. So just like you have it, you are going to make use of the two before accession numbers. That is what you are going to do. You do four accession numbers. The first one belongs to Pseudomonas aeruginosa, which is mainly used and mainly common in polluted water, and with the accession number already displayed for you. Then the second one, you are going to make use of Acinetobacter, and that's the accession number is also um, displayed. Then you also make use of that of from clean water, which is from stream B. That's going to be Nitrosospira multiformis. So the accession number is displayed there. Then you carry out blast on it. So that is going to be what you do. Then there is going to be step-by-step analysis of how can you retrieve DNA sequence from NCBI, just like you know, and the step is already displayed as you have it there. So don't forget, we are going to deal with four different organisms with four different accession number, and the accession number is actually displayed as you have it here. So just like I said, there will be time for practice activity, and you'll be able to now utilize ChatGPT to be able to also do the analysis of your results. So, and after that, the next activity after that is called DNA barcoding. DNA barcoding, which is a method used to identify species using short standardized DNA sequence. So and barcoding regions that we are actually looking for include COI, RBCL, MATK, 16S ribosomal RNA. We are going to make use of this as an activity in the class. Then we are going to use another one that is going to make use of that means we are going to make use of three. So use one in the species, you have the ID number, and the one that you have in barcoding. So barcoding is always used for species identification. It is also used for biodiversity studies as well as ecological monitoring. Okay, and the different types of databases we are going to make use of today is normally the NCBI stroke, the gene bank. Then you make use of BOLD, which is a system that where you can get the barcode from. You make use of BLAST. You are familiar with Mega as well as iTOL. We are going to make use of all these when the time